might have to minimize your PowerPoint to see it. Get that. Good. Zoom.
Is it okay? Yeah. Okay, so hello everyone, my name is Larissa, and today I'm going to present my work on the function of RET in the crosstalk between pancreatic ductal adenocarcinoma and sensory innervation. So first, I'll give you an introduction on pancreatic ductal adenocarcinoma, or PDAC, as I will refer from it from now on. So PDAC is a highly aggressive form of cancer with poor outcomes and also a rising incidence. It is one of the major causes of cancer-related mortality, and here in Canada, PDAC is the third cause of cancer-related mortality. There are several reasons behind the high mortality rates associated with PDAC, and one major challenge is the late detection of this disease. Early symptoms of PDAC are non-specific, which means that the diagnosis often occurs when the cancer is already at an advanced stage, marked by the presence of metastases and pernary invasion. Detecting PDAC at such a late stage significantly reduces the chance of successful treatment and also contributes to the poor response to the current therapies. However, it is important to also remember that a tumor is more than just cancer cells. The tumor microenvironment includes various elements that influence how the tumor grows, invades, and also surrounding tissues. These nerves, and in this context, a key focus has been on the role of nerves within the microenvironment. These nerves have the potential to influence the progression of the tumor, for example, by secreting neurotransmitters, neuropeptides, and growth factors. Therefore, understanding these interactions is essential to gaining insights into how a tumor behaves and how we might develop more effective treatments. In solid cancers like PDAC, there are various ways in which the cancer spreads. This includes invading nearby organs, vascular invasion, invasion through lymphatic vessels, and also invading nerves. As I mentioned earlier, pernary invasion, or PNI, is a significant characteristic of PDAC and contributes to its aggressive nature, which occurs when cancer cells invade and grow along nerves, facilitating tumor progression and metastasis. PNI is considered a poor prognosis indicator, not just in pancreatic cancer, where it's present in over 90% of patients, but also in other cancers like head and neck, prostate, breast, and colon tumors. The mechanisms involved in PNI include interactions of cancer cells with chemoattractants derived from the nerve environment, such as neurotrophic factors, which lead to migration and invasion. In addition to its role in tumor progression, PNI also contributes to the occurrence of abdominal pain in patients with PDAC. Additionally, when tumor cells invade nerves, they can disrupt the normal structure of the nerve's protective shell, leading to the release of inflammatory substance, but resulting in pain. So understanding these processes is vital in managing and treating cancer. Another characteristic of PDAC that I would like to highlight is the presence of RET expression in a significant proportion of the PDAC cases, going from 40 to 65% of the patients, especially in high-grade tumors, which are associated with pernary invasion, PNI, poor prognosis, and also reduced overall survival. The RET product oncogene encodes a receptor tyrosine kinase that it's essential for the development of multiple systems, including the nervous system, kidney, and endocrine organs. In the nervous system, RET is critical for the development of the enteric nervous system, which regulates gut motility as well as for the sensory innervation development. RET activation in the cell membrane is mediated by its interactions with GFLs, including GDNF and arginine. GFLs will interact directly with RET receptors, known as GFR alpha 1, and GFR alpha 3, in this case, GDNF will bind specifically to GFR alpha 1, while arginine will bind to GFR alpha 3. In this study, I will focus on these two co-receptors and its ligands because they have been shown to be highly expressed in PDAC tissues. This specific binding, of ligands to the co-receptors will result in the creation of the GFR alpha red complex, facilitating the autophosphorylation of intracellular tyrosine residues. And after that, red will be recruited to the lipid rapids for further signaling and also internalization, both of which are critical for the activation and regulation of red signaling pathways. This ligand receptor complex activate downstream signaling pathways, including PI3K-AKT and MAPK-ERK pathways, which will influence cellular processes such as survival, differentiation, proliferation, migration, and derived outgrowth. So as we can imagine, alterations in red function can lead to its oncogenic behavior. This disruption can occur through either gain of function mutations or fusions, which result in the constant activation of the red receptor or an increase in the expression of the wild type receptor. As I mentioned before, increased expression of the wild type red receptor has been observed in various human tumor types, including PDAC, where it plays a role in tumor progression. As shown in this figure, the activation of overexpressed red plays a role in promoting tumor invasion, proliferation, survival, and also metastasis. So far, we've explored the cancer side and the mediator side of this interaction. And now we need to go a little bit into more details to the other component, which is the innervation. I've previously emphasized the significance of PNI in the context of PDAC progression. To understand the process involved in PNI, it is also important to explore how the pancreas is innervated and what changes occur when these nerves, when cancer begins to develop. So the pancreas is connected to a complex network of nerves that are crucial for its function. In this figure, we can see that the pancreas is innervated by sympathetic nerves, parasympathetic nerves, and sensory nerves. In the context of PDAC, the innervation of the pancreas becomes even more significant. Studies have shown that PDAC is associated with extensive innervation, which can have both tumor-promoting and tumor-suppressive effects. Studies from various research teams have suggested that the sympathetic and sensory innervation can promote the growth of pancreatic cancer, while the parasympathetic system appears to limit the development of tumors in the pancreas. So building on this knowledge, a study using an in vivo model of PDAC has demonstrated that the target elimination of sensory neurons effectively impaired cancer initiation and progression. In this research, the authors used capsaicin, which led to the elimination of the sensory nerves in the pancreas. So in the image A here, we can see a tissue without capsaicin administration, and we can see the presence of nerves indicated by P. And in B, after capsaicin administration, there's no presence of sensory innervation. So they demonstrated that animals treated with capsaicin had a delay in initiation and spread of cancer. And here in image D, we can see a survival curve showing a significant improvement in the survival of these animals. So in red, we have the curve after administration of capsaicin and without sensory innervation. Supporting this other study showed that when PDAC cells are co-cultured with DRG sensory neurons, there was a noticeable increase in their growth and survival compared to those cells on their own. As we see in this graph, one of their experiments, PDAC cells had a greater colony formation, which is a measure of cell proliferation when they were co-cultured with DRG neurons. Furthermore, interesting evidence comes from also from studies with patients. So research found that patients with gastric cancer who had their vagus nerves removed during surgery had better results when compared to those who only had the surgery to remove the tumor. And together, these studies highlight the critical role of the interaction between sensory nerves and tumors in driving disease progression. This not only opens up an interesting path for potential treatments, but also stresses the need for better understanding of the exact mechanism governing these interactions. And as I mentioned earlier, the involvement of RET is also important for the neuron's development and maintenance, especially within the sensory nerve population. In this table, in this figure, we can see distinct populations of sensory neurons highlighting RET expression. So we have RET being expressed by different populations of sensory neurons. And RET is expressed in about 70% of the sensory neurons. Therefore, given the importance of these neurons in cancer progression, exploring the role of RET in this complex interaction holds a substantial potential. So from the perspective of cancer researchers, RET is known to mediate critical processes that underlie tumor invasiveness. This can include functions like cell polarization, migration, invasion, and also the expression of metalloproteinases. On the other hand, when we view from the neuroscience perspective, RET, with a particular focus on GDNF and GFR-alpha-1, plays an important role in promoting neuronal survival, supporting differentiation of neurons, and also facilitating neuroid growth. These are essential functions for the development and maintenance of the sensory neurons. What's intriguing is that there's still a lot to uncover regarding how RET may mediate the interactions between these two distinct cell populations in the context of PDAC. With that in mind, the main question we are addressing in this work is, how does RET impact the communication between cancer and nerves? This process is complex and probably involves communication in both directions and a mutual attraction. Some evidence have shown that neurotrophic factors such as GFLs have been identified as overexpressed in PDAC, and their upregulation has also been linked to enhanced nerve growth. Lastly, beside the neurotrophic factors that can be released by neurons and pancreatic cancer cells, there's another part of this communication system. There is some evidence suggesting that the release of the co-receptors, the GFR-alpha co-receptors from nerves in the tumor's environment. So this release helps the nerves and pancreatic cancer kind of to talk to each other. On the flip side, pancreatic cancer cells can release GDNF, which helps them grow more by making nerves fibers develop and by guiding axons. It's almost like a sign that they have to direct the cancer cell migration. So all these pieces together suggest that the GFR-alpha RET system could mediate cancer and nerves interaction and causing the nerves to be invaded. So in summary, since most of the PDAC cases are detected when they have already spread and PNI is present, and also considering the elevated RET expression in PDAC tissues, and also its correlation with PNI, as well as its functions in cell migration and invasion, along with the significant role of RET and its ligands in promoting neuron outgrowth, it becomes crucial to comprehend this mechanism and how it could mediate the interaction between cancer and nerves. So our hypothesis for this study is that the activation of the red receptor by neurotrophic factors released by sensory innervation promotes the invasiveness and metastatic potential of PDAC cells through the induction of cell extension formation and migration, while PDAC cells modulate sensory neurons to promote tumor-supportive neurotrophic factor release. So for testing our hypothesis, we have three aims as shown here. The first two aims will concentrate on assessing the effects of RET activation on each cell type, while the third one will assess their combined interaction. So aim one will explore the role of RET in mediating the effects of sensory innervation on PDAC cells. And you'll see these little figures throughout my presentations to help you to follow along. So aim two will evaluate the impact of RET activity triggered by PDAC cells on sensory nerves. And finally, aim three will focus on characterizing the RET-mediated interaction between sensory neurons and PDAC cells. First, I'd like to define the experimental model that will be used in this study. The initial component will consist of PDAC cell lines, specifically Miopaccus and PANC1s. I'd like to emphasize some things that influenced our choice of those two cell lines. Both cell lines express the RET receptor, as we can see here. PANC1 cells exhibit higher levels of expression when compared to the Miopaccus 2, but both express RET and GFLs, GDNF. Additionally, both cell lines also express the co-receptors GFRF1 and GFRF3, along with their ligands, GDNF and artemisinin. And as a result, the wild type RET expressed in these PDAC cells is consistently activated at low levels due to autochronous signaling. To investigate RET functions with the cell lines have generated CRISPR RET knockout Miopaccus cells, and we are currently in the process of making PANC1 RET knockout cells, and we are also working on selecting the guide RNA for the GFR-alpha-3 knockout cells, which will be completed soon. Furthermore, in some of our experiments, we also used a sapercatinib, which is a specific RET inhibitor. Another critical aspect of our experimental model involves the primary culture of DRG neurons. Our first step was to establish a primary culture of these neurons. This involved the extraction and isolation of DRGs from a mouse spinal cord, which was kindly done by students in the LAMAX and Tablet lab. After isolation, the DRG were cultured in a plate coated with polyol-lysine, and we could then observe their progression over time. In this image, you can see what healthy neurons should look like. They appear round and well-defined, and they have well-defined borders. In image 8, we'll observe the initial state of the neurons at the start of the culture, which shows no significant neural outgrowth at this point. After 24 hours, you can see the early signs of the neurite outgrowth, indicating the beginning of this process. And then finally, after 48 hours of culture, we can observe further neurite outgrowth, showing us the ongoing development and maturation of these neurons. It's also crucial to highlight that the DRG culture comprises a complex mixture of different cell types, including neurons and non-neuronal cells, such as glial cells and fibroblasts. Therefore, we will also employ specific markers to differentiate the cell types, which will be also available for this study. One of the tools we've used is the NEWAN antibody, which labels the nuclei of neurons. So as we can see here in this figure, the NEWAN probes to be available for distinguishing neurons from the non-neuronal cell types present in DRG culture. So here we have two neurons, and their nuclei is marked by NEWAN, and then we have many other cells around, which are the glial cells and the other non-neuronal cells in the mixture of DRG neurons. So the first aim was to explore how RAD mediates the effects of sensor neurons on cancer cells. Previous finds from our lab have indicated that RAD activation following treatment with GDNF results in in cell polarization, cell invasion, and the formation of invasive structures. Based on these results, we are interested to look at the specific impact of GFR-alpha-3 and artemin in these mechanisms. Additionally, we want to determine whether the secretome of sensory innervation can replicate these effects. So to accomplish this, we will conduct cell polarization, individual cell migration, cell invasion, and gelatin xenograph assays. Our initial result with PANC1 cells post-treatment with GDNF demonstrated the constitutive activation of RAD, leading to the corresponding constitutive activation of downstream pathways, as we anticipated. So moving forward with this aim, our next steps will involve performing signaling plots with all the cell types I mentioned earlier, and that will help us to illustrate how manipulating RAD or its co-receptors can impact the downstream pathways. Subsequently, we'll investigate whether artemin, GFR-alpha-3, or DRG conditional medium can contribute to the invasive phenotypes induced by RAD in cancer cells. To assess the influence of these conditions on single-cell migratory capacity, we will use the InQSight system for a cell tracking assay. This will allow us to quantify cell movement, helping us to determine if RAD activation under different conditions affects individual cell motility. Additionally, we will employ a healing assay to study the impact of RAD activation facilitated by the same conditions I mentioned before on the collective migration of PDAC cells. Also expanding upon prior findings, where we observed that the invasion of PANC1 cell spheroids was inhibited under conditions of RAD inhibition, even in the presence of DDNF stimulation. We will investigate whether the effects seen with RAD activation by DDNF can be replicated with DRG conditional media or artemin. To assess this, we'll use spheroids embedded in a collagen matrix, and then we'll measure invasion using area measurements. Our hypothesis is that the spheroids will respond to the stimuli provided by either DRG conditional medium or artemin, and that the spheroids made with RAD or GFR-alpha-3 knockout cells will have less invasion. Following that, since metalloproteinases are also key contributors to matrix degradation, which is an important mechanism for cell invasion and tumor progression, we'll quantify and we'll evaluate MM2 and MM9 activity in response to the same conditions after DRG conditional media treatment and after artemin treatment. So with this, based on our hypothesis, we expect that PDAC cells treated with DRG conditional media will activate RAD and then induce matrix degradation via metalloproteinase activity. Our next objective is to understand how RAD activation mediated by PDAC cancer cells, can influence DRG neurons. So as I mentioned before, there are different populations of sensory neurons expressing RAD. However, it remains unclear whether this effect is specific to a subset of sensory neurons within the DRG population. So we want to evaluate the specificity of RAD activity in promoting neurite formation by treating isolated DRG neurons with DDNF, artemisinin, and saprocanin. By including artemisinin in our experimental design, we aim to understand whether the observed effects on neurite formation are specific to the different GFR-alpha receptors. So the isolated DRG neurons will be were cultured in F12 medium, supplemented with DDNF, artemisinin, or saprocanin. Here I only have the data plus DDNF and saprocanin. And after isolating and treating the DRG for 72 hours, we stained the DRG neurons with calcium red orange, which is a dye that will mark all the live cells. Here in this image, you can see a representative pictures of these DRG neurons. So this is our control after treatment with GDNF and then after treatment with saprocanin. After analyzing the treated DRG neurons, we saw that GDNF treatment didn't have a significant effect on axon outgrowth, neurite length, or on the number of branching points compared to the non-treated group. However, when we treated them with saprocanin, we noticed a significant difference in the neurons' morphology, and they had smaller neurites and a decrease in branching points. This suggests that RAD inhibition may have an impact on the shape and structure of these neurons. Here we can give a better look at these neurons treated with saprocanin, and it's quite evident that after RAD inhibition, the neurons displayed shorter, thinner, and more numerous extensions. So we can see all they're really small and they have much more when compared to our control condition. And this strongly indicates that RAD plays a significant role in shaping the complexity of these axons, of these neurons. So also to explore whether various populations of sensory neurons respond to RAD activation, we intend to use DRG neurons from TD tomato, for example, mice, transgenic mice, to identify a specific subset of sensory neurons and how they respond to RAD activation. These mice will be kindly provided by Dr. Sebastian Tabo from the Department of Biomedical and Molecular Science sequence. So to assess the impact of RAD activation on the neuron complexity, we will measure neurite number, longest neurite, total neurite, and also the number of branching points. To expand our knowledge of how RAD activation may influence sensory neuron function, we will also investigate its potential impact on synaptic modulation. Following the culture, the 72 hours of culture, we will fix the DRG neurons and we'll stain them with two markers, one for inhibitory synapses and another one for excitatory synapses. And with that, we aim to visualize and quantify any alterations in the distribution and abundance of inhibitory and excitatory synapses in response to RAD activation or inhibition. Lastly, for our aim 3, after demonstrating the isolated RAD functions on PDAC cells or DRG neurons, We aim to provide a comprehensive characterization of the RAD-mediated interaction between sensor neurons and PDAC cells. By employing co-culture and 3D models, we will investigate key aspects of this interaction, including cancer cell migration and invasion toward DRG neurons. We will evaluate the migratory capability of PDAC cells in response to direct co-culture with DRG neurons using a modified migration assay. And also using an indirect co-culture model, we aim to investigate whether PDAC cells can migrate towards DRG neurons. In this image, we have a representative picture of the cell migration assay that I'm currently optimizing. So on the left-hand side, we can observe the DRG neurons, and on the right-hand side, no, it's opposite. Here we can see the DRG, and in this side we can see the PDAC cells, which I was working with the MIA-PACA2 cells. So the primary goal here is to establish a timeline going from 0 to 72 hours, which will enable us to see the migratory ability of cancer cells, and potentially we could see the directional near-right outgrowth towards the cancer cells. Next, we aim to determine whether cell functions mediated by RAD, such as cell invasion, could be activated through co-cultivation with DRG neurons. So to assess the ability of the PDAC cells to respond directionally to chemotrectants released by the DRG neurons, we did a transwall invasion assay. In this assay, the upper part of the membrane was seeded with either MIA-PACA2 RAD knockout or a wild type, while the bottom of the plate was seeded with HAKs293, which were our negative control, SMC cells, which are known for secreting GFL, so it was our positive control, and then our DRG neurons. Our results showed that MIA-PACA's RAD knockout, as we can see here, they had a lower number of invading cells compared to the MIA-PACA2 wild type cells in response to any type of cancer of cells seated at the bottom. And interestingly, we saw that MIA-PACA cells displayed higher invasion rates towards DRG neurons and SMC cells when compared to the HAKs. This findings suggests that DRG neurons may have a stimulating role in the invasion of pancreatic cancer cells, likely due to the secretion of chemotrectants molecules. Finally, this work also involved optimization of co-culture experiments to explore how these different cell populations interact with each other. First, to investigate the role of RAD in the interaction between nerves and PDAC cells, we co-cultured DRG neurons and MIA-PACA cells. And these cells were co-cultured for a period of 3 days. And after that, we assessed the length of axons and also the number of branching points. What we found was that there was a noticeable change in the length of the longest neurite, which was increased, and the number of branching points, which was decreased after the co-culture with the MIA-PACA cells. These observations can suggest that the neurite outgrowth appeared to be more directed towards the MIA-PACA cells, indicating a potential role of RAD in mediating this directional response. in conclusion, the presence of Eopaca cells, which are a source of GFLs, GNF, likely influence the directionality of neurite outgrowth towards these cancer cells. So this can be a sign of the significance of GFL's red axis in facilitating the crosstalk between the cancer cells and nerves. So after that, in addition to investigating cancer cell migration and invasion, we will perform a direct co-culture assay to assess the invasive capacity of PDAC cells towards the DRG neurons. By culturing PDAC cells and DRG together, we'll be able to quantify the extent of cancer cell migrations and also the extension of neurite outgrowth. So we'll also perform a 3D model where we'll be able to visualize the cancer cells migrating towards the DRG. So you basically form like a spheroid with the cancer cells, and the DRG neurons, they also aggregate really easily. So you can have pretty much two 3D structures, and then you can see the migration of cancer cells through this bridge, something like that. And this will enable us to capture a real-time, I want to do like a real-time experiment, and again, some insights into the dynamics of cancer cell migration towards the DRG neurons. And I will also do the same direct co-culture experiments using the other cell that we are going to generate. So by considering both aspects of the crosstalk, this project will investigate bidirectional communication between PDAC cells and sensory neurons, uncover a potential positive feedback loop that may fuel tumor progression. And understanding how PDAC cells modulate sensory neurons and how sensory neurons in turn impact PDAC cell behavior will be crucial for developing comprehensive strategies to disrupt the crosstalk, potentially leading to novel therapeutic interventions for pediatrics. Thank you so much. I'd like to thank my supervisor, Dr. Liz Milligan, and also my supervisory committee members, Dr. Andrew Craig, Dr. Chris Nicol, and Dr. Peter Green. Thank you.
So, very nice talk.
Thank you.
Ready for some questions now?
Yeah.
You want?
What would happen if you were to have a mouse with, say, PDAC, if you were to give it a RET inhibitor? What would happen? That,
to the nerves or?
Well, to the tumor and just, well, the whole idea being, you know, that the crosstalk between the two has implications to the tumor biology. Would you expect to have effects on tumors in vivo or maybe on their metastasis, or what would that look like in vivo?
Yeah, so that's a good question, because the RET inhibitor, it is more specific to, it has been used for some tumors in case when they have a mutation or like fusions. And in the case of PDAC, the functionality of RET is the wild type RET. So it is overexpressed. I would say that we would see a decrease in tumor for those patients too, for those animals too in the case. I don't know about the nerves, like how could subretinib affect nerves in the in vivo context. I haven't read anything about that, but by everything I've read and seen all the facts, the GFLs have in the development of nerves, innervation, and also in the cancer, I'd say that we might see also an impact in the innervation of those tumors.
So have you tried that inhibitor in any of your cell models, your co-culture systems, for example?
Not yet.
No.
Yeah, just like I tried superkinase. I treated the neurons with superkinase, but it was only the neurons.
But can you block autophosphorylation?
Mm-hmm.
So you validated it actually works.
Yeah.
Ret. Okay. Good. Other questions? Dr. Craig?
Great presentation. Just wondering if you thought about, in the context of the tumor microenvironment, just kind of picking up on the box question, are there any other cell types within the stroma or tumor microenvironment that might also be contributing to potential crosstalk between red and some of the nerves?
Yeah, macrophages could also be involved in this process, since I've seen some studies that show that they also secrete GFLs, so they also could play a role in this interaction. It's kind of simplistic to say that it's only tumor and nerves. There's so many more players in this communication.
I think these tumors can be quite fibrotic as well. Have you thought at all about cancer-associated fibroblasts?
Yeah.
Reason to consider that either as sources of signals for crosstalk or ways to.
Yeah, I know that they also have they also can impact this interaction, but I found already hard to understand the interaction between two players, neurons and cancer cells. So I'm not sure how we could add more players to this scenario. No, but yeah, they're also important. They do have a role in this interaction too.
Thank you for this really interesting presentation. First of all, your figures are spectacular. I really thought those slides were quite something. Anyway, this is such an unusual thing. I've never really heard about this before. So if I'm allowed 3 very quick questions. What percentage of the progression is through perineural versus local, vascular, and lymphatic? Is this a major contributor, or is it any idea?
So just for PDAC?
Yeah.
So for PDAC, almost more than 90% of the patients, when they are diagnosed, they have PNI. So this perineural invasion is present.
Second question is, what the heck do you need sensory nerves to your pancreas for? I mean, I don't get that. Some of you must be neurobiologists, but why do you need sensory innervation of an organ? so that follows on my last question is, why don't you ablate the dorsal ganglia? Has anyone done that to see whether that would influence the
cancer?
Yeah.
Yeah. So there are some studies that they tried the innervation of the tumor, and they saw a regression in tumor progression after denervation.
Because you showed that those capsaicin results on neonates.
Mm-hmm.
But if you've got an established pancreatic cancer, I have no idea whether that would.
They also, there's many data saying that they see more nerves in the cancer tissue when compared to the normal tissue. So it is something that they've observed, that there's more presence of these nerves, and mainly sensory innervation after cancer progression. And yeah, I've read some papers when they say when there is denervation, the cancer tends to not progress.
Even that's why we need sensory nerves.
Okay, so the focus of this, so we have the central system, nerve system, and the peripheral nerve system, which are important to basically do the communication between your pancreas and your brain.
What does a pancreas have to say to a
Well,
brain?
I'm sorry. The brain is active. I don't like the hate pancreas. Can you?
Yeah, I mean, sorry.
Thank Dr. Lillicrap for, is it Dr. Alan Lomax that you collaborate with?
Yeah.
Okay. In the future, you might want to fix it says Dr. Alex.
Okay.
So
Oh, yeah, sorry.
Dr. Lomax looks at the enteric nervous system. Is that involved at all in the pancreas? Because the pancreas goes into the small intestine. I'm wondering if enteric nerves might be a contributing pathway.
Yeah, that's not.
Because you get a lot of spread into the small intestine from my very rudimentary understanding of pancreatic cancer. But.
Yeah, that's not exactly what I'm focused on, but yeah, it can for sure have an influence on the invasion, the way that the cancer cells invade.
Any other questions? I don't see anything in the chat, and no hands are up in the Zoom audience. If no other questions, then join me in thanking Larissa for a really nice talk.